and Luthey Schulten is going to tell us about simulations of cellular processes.
Okay, since we're giving many talks, I can only give you one or two examples, but these examples that I'll mention in my talk were the posters that you see outside. I think they're still up, so if you have additional questions, please ask the students who are there. And what I want to show you is how we're developing techniques to look at cellular processes at a whole cell level, as well as on the colony level. And to do this, we've had to sort of use a different methodology for treating things that is sort of commonly known in biological physics. So I know everyone's familiar with molecular dynamics, bounding dynamics, but they have some limitations on what length scales and time scales they can reach. To begin with, the time steps are like femtoseconds or picoseconds, and people are doing really heroic work to get up to the millisecond level. Where I want to be is to go over and work at the whole cell level. I want to look at reactions, and I want to be able to go up to hours. And so we will be using time steps of microseconds. And the thing is you look, as you go from the same problem, this is DNA and ribosome, in this direction, we've had to give up resolution, but we gain in being able to do reactions. Also, the description becomes probabilistic here. And you also see that I've imposed a grid on it. So it's important when you're doing it at the single cell level, the performance and what you can do depends on what's the fastest fusing particle in your method. But everything I'll show you is typically from like 16 to 32 nanometers at the single cell level. All right, so what does it mean to be the probability of a cellular state? So this P and the X zit represents everything that's in the cell and the description of the biological process you're trying to model. That could just be ribosomes, it could be messengers, it could be proteins, it could be DNA. And if the cell were really well stirred, and particularly if it's involved in gene expression, you almost can't say that phrase without saying stochastic gene expression. Because you're going to have a mixture of small numbers, large numbers, but you could use sort of a very simple chemical mass equation where you just look at the probabilities that take you out of that state or into that state. Unfortunately, the cell is not well stirred. It's very noisy. This is more realistic. This picture of the iPoLS cell is based on a tomogram that we got from Wolfgang Baumeister's lab in Munich. And you can see here, the shaded area in the middle is where the DNA would be. The ribosomes we got from their cryo-electron tomography reconstructions. And you can see a gene going off and on here, and when it's on, it's making messenger. And then it makes a protein that goes to the surface here and moves across the surface. So there's a lot of dynamics involved. And if you're going to do a realistic and a good job of modeling cellular reactions in the cell, you really have to use all the available information that's out there. So architecture we can get from cryo-electron tomography, dynamics, diffusion coefficients we can get from people like Sanshi, Johan Elf, Werner, Tinker Ha, and models for kinetics we turn to people like J.D. Williamson and my other colleague at UI, Tom Kuhlman. So now to put in the spatial heterogeneity to this well-stirred approximation, you have to add diffusion. And if you look, there's an additional indice now, because I have to talk about what's going on in each sublattice. And I want to remind everybody, this cell is really packed. This is about 50% packing, and you can see there's still areas there, but it is quite a crowded environment to deal with. So the two problems I'll talk to you about, this is where we started to first calibrate our work. This is on a stochastic gene expression. There's a lot of data on the lab genetic switch. We can check our methods. You see a little clock ticking here. This was a simulation up to about 56 minutes. And to be able to get to that time scale, we have to do everything that we've developed, these labs micro software on GPUs. This was relatively small for what we do today. This had only like about 24 reactions, 10 species. When we do ribosome biogenesis, we're up to thousands of reactions, 251 species. And then if we go up to the colony level, well, it just expands enormously. And we'll do a slight shift in our methodology here. We become even more coarse-grained up here. But what we add to it, we can look at the environment around the cell. the microenvironment, see how it affects each cell, what it takes up, whether it's taking up, I don't know if you can read this, is oxygen, glucose, and what it is secreting. All right, so just to reiterate, we try to put in molecular crowding, so that means we have lots of information on protein and RNA distributions. If you work on model systems, because if you do E. coli or yeast, there's lots of that data available. Most of the reactions will be described by this reaction fusion master equation. We're using our software lattice microbes, genetic parameters we get from a number of places. From biochemistry, 20 years or so has been invested in getting rates for those single molecule experiments. And then, as I said, we can look at whatever process we want, like whether it's monitoring small RNA production, or in our case, I'll talk to you about ribosome biogenesis. To go up to the colony level, we do sort of a hybrid method of our RDME method, but we have to, in order to put in the whole network, we have to be able to treat the fluxes through all the reactions at a steady state level. So it's going to be iterative. A certain amount of time is spent doing reaction diffusion, and a certain amount of time is just calculating the fluxes and the growth rate and the expansion of the cells. So it's really these last two papers that just appeared this year that I'll be talking to you about. Now, if you're in Illinois, you always have to work on the ribosome. And in fact, my interest started on it because of my colleague Carl Bowes. With him, I started a paper in 2008 to look at the signatures that identify the ribosomes from the 3 domains of life. As you all know, those are used now to classify all organisms. We wanted to know what those signatures did, so in a series of papers on molecular level, and the latest one just appearing with Sarah Wilson and Jacob, was looking at the assembly of the ribosome. I forgot to have this movie play, which is just so pretty and important. So that's a beautiful object, worthy of 3 Nobel Prizes, I guess. And so let's talk just about assembling of the small subunit here, where you're seeing the yellow and the brown. So the brown are the proteins, the yellow is the RNA. So what if you want to assemble that object? There's one 16S rRNA, but there are about 20 proteins. In principle, if there weren't some order to it, there could be 2 to the 20th states. But we know already since about 1977 from Nomura's experiments that there's a hierarchical assembly of it from the 5' to the 3' end, and the proteins have to first come and bind to the RNA directly. Those are the primary proteins, and these guys bind to those, and those guys bind to those. So if you start off with the RNA and you throw in the proteins, and say one of them is a secondary protein, it shouldn't bind. And that will give you a reduction of those states to about 1,600 states. The only additional complication you have is that in E. coli, it has 7 of these rRNA genes. But we do know from both Baumeister's cryo-electron tomography, some labeling that Sunny Shi did on the ribosomes, and as well as Tom Kuhlman, that this is about what the distribution should look like. And for slow growing, we know the number. So from the pulse chase experiments of J.B. Williamson, who's a co-author of this paper that's just appearing, we could get binding rates of all those proteins to the 16S. There's a 1,600, but even that was too many for us to treat initially. So we tried to reduce the number of intermediates we considered. And you're not supposed to read this, it's not an eye test. It's just to show you start with the negative 16S and you end with the 30th, small subunit. And what we did, and you see the lines, some are much thicker than others, those are fluxes. So all we did is we eliminated those intermediates that didn't contribute much or have much flux or much occupancy. And the student doing that work was Tyler, as it's posted outside. So that model that we did, using J.B. Williamson's data at 40 degrees Celsius, we put into our in vivo model of biogenesis, ribosome biogenesis, along with the messengers, the ribosomal proteins, and we started off with transcription, and translation, and then assembly, and we followed it over 120 minutes of the cell cycle. We started at a sort of steady state situation, like just forming the cell, but then we turned off initially the 16S. We just wanted to follow and allowed it to start. We wanted to really look at the making of the new small subunits. So what you can see here, if you look at these lines, over the time course of the doubling time for the 120-minute doubling time of this bacteria, the number of ribosomes goes from roughly 3,000 to about 6,000. And we compared this then with also work that was done with Tom Kuhlman. This is work that we're just trying to get out, where he actually could measure the number of ribosomes coming off of each of the 7 operons. And this is a function of the doubling time, so we're sitting right about here. Here's a blow-up of it, where he compares the storm results with qPCR. So again, we get about 6,000. We sort of tried to set our parameters to more or less see if we could reproduce this, and the answer is yes. And now what we're trying to do is look at storm data and try to understand the spatial organization of the ribosomes and why do they occur where they occur. Now to do this, those calculations I just showed you, those took us 3 days using a special code generation called just-in-time codes. And it's really important, it's just like somebody doing a new experiment. You cannot go to these kinds of processes, the universal processes, without investing a lot of effort in the computational methods that you use. So when we started off last year in January, and this is the simulation volume. I've marked here E. coli, divided E. coli, yeast, and red blood cells, systems that we were considering. The vertical axis here tells me how many days I need on the computer to get 1 hour biological time. And we were just making a day to do an hour of E. coli. And now when we switch to this new GPU, and can also use, if the system has multiple GPUs, the curves also improve. So the red line would be, say, for one GPU with 16 nanometer lattice size and a 50 microsecond time step. You can see that you're under a day to do for this benchmark reaction. If you do 1,000 reactions, it takes a little bit more time. And as I say, you can get it done within 3 days. If you're interested in using the code, and more people are using it, and we do have them on our website, we give it out, just like we do VMD and NAMD, along with a set of tutorials. Now, what I told you about was just this part. These were all reactions that go on in the cell, and so let's zoom in a little bit more. There's translation. Well, what about the rest of the reactions? What about metabolism that I left out? And some of the signaling. Well, we're looking now how to put that in there too. And once we have this united with this model, then we've got a complete cell simulation from fundamental principles. So to do this, I first put in the reactions. This is like a simplified diagram of the core metabolic map of E. coli. What we considered first was a colony of cells, because then we could treat the microenvironment differently of here. And what these guys on the outside might see more oxygens, and the guys in the middle, that's going to impact what parameters come into the system. Also, they're going to see different foods depending where you are, and depending on the state of your system, you're going to secrete different things here. So acetate is going to play a role. And how did we do this? We actually did both a simulation first, and then we checked it with an experiment. So we grew the cells from a single cell on an arbor plate, and in the computations, what goes on is you have a diffusion event, and you look at all the particles. This could be oxygen, it could be glucose, and then that tells you the input into your steady-state analysis of the flux and the growth coming out from your network, and that growth pattern is fed back. So now these cells are growing. When do they expand into the next lattice site? And I think it's clear, now this lattice site is no longer 16 or 32 nanometers, it's 10 microns. So each of our lattice sites now contains several bacteria. And that controls how quickly they expand into the X, Y, and Z direction. And you keep doing cycles of this, and we grew it up to about 36, I think some of them 48 hours. And what did we see? Well, this is from the simulation. This just gives you the growth curve as a function of time. These are more interesting, but I'm afraid they're too small for you to see. I'll tell you, this is oxygen concentration, and this is glucose concentration. The bottom part is always the agar. So you can see after a while, the guys in the center of that colony get depleted. And the same way, here's the glucose. Everybody's happy at the bottom. They're getting lots of glucose from the agar because there's no glucose in the air, right? So, but then you see a change in all these metabolites develop a gradient. What's interesting is our model. When we compare it to the experiments, and here we put in a plasmid with a reporter system, and what we were reporting on, the promoters, were at places in different parts of that network. So we knew when does this pathway get turned on, when does that other pathway get turned on. And one of them, the points, the lines are from the theory, and the dots are from our examples at different time states. And you see they agree pretty well. The only thing we changed here, well, is how much packing do we allow in any sublabs before it goes off into the neighboring cells. The only thing we missed is this transition from 2D to 3D. And the experiment that we did to validate these simulations, which I keep pointing out were done first, this is after 30 hours, we really saw that those gradients, they were driving metabolic differentiation inside the colony. That you had, they developed what you call acetate crossfeeding. So the cells here at the top, they start consuming acetate, and the cells at the bottom here start producing acetate, and they consume glucose down here. And to be able to see this spectroscopically, we put in a two-colored plasmid, one with the I think it's M-cherry, for this last step here, this PTSG, that gets you into the glycolysis pathway. And then we put one for one of the genes down here where you secrete acetate. And here are the results, so let me go through. We then did the optical sectioning and looked at colonies, sort of cells at the top of the colony. So you saw only the green dye being turned on. Then as we went further down, closer to the agar, you now saw the M-cherry signal coming in. And here on the side, we're showing you a vertical slice through. So it agreed beautifully with the theory. Now, when I say beautifully, I haven't decided to skip the conclusion and said, just show you the people who did that work. So the people that were involved with all the ribosome work are sort of clustered over here. And the people that are involved with solving steady state and the cell and colony simulations are here. And it's very important also our outstanding programmers who help speed up our method. I think you also heard John's talk on putting in the effects of our DNA replication and gene copy numbers into the simulations. That we're doing presently, and we're hoping it'll be quite straightforward. And with that, thank you for your attention.
Questions for Zan?
Do you believe you can run parts of these networks, other parts, like for example, I'm just doing genomic part, I'm not doing metabolism. How to run only these parts, the other parts, not doing they're all coupled together, right?
Okay, so that's a good question. Like, why can you treat some things sort of modularly? So now the whole field of like...
yeah, but you should learn about modularity, right? You shouldn't sort of impose modularity. How much you learn, how much you impose? That's the question.
Well, okay, so like the example that we did on the metabolic pathway, coupling it in the colony, was in part, had two goals. One was to check the metabolic network. That is the most studied system out there, is E. coli in the network properly. And we were then time-dependently updating it. What we didn't do is do the change of the ribosomes in there. Will that have an effect? Yes. But there are certain timescale separations that do allow you to treat parts of the network in one time regime, translation in another one, and sort of weakly couple them. Now, there are already steady state models that attempt to do that. We just want to do it now totally dynamically because we think they're missing some things. And that'll be our goal. And I just put up a movie. This was the simulation just so you can see how it really takes place.
More questions from the audience? Okay, if not, let's thank Zan again.